Howdy, it's Mr. Justice. In this video, we're going to go over the details of cell respiration. We'll talk about how chemical energy in food can be transformed to ATP. We'll learn a little bit about mitochondria and cellular respiration inputs and outputs. And then finally, we'll explain the functions of glycolysis, Krebs cycle, electron transport chain, and ATP synthase. So let's get started. One of the questions that we asked in class, what did you eat for breakfast? A lot of students mentioned that they ate like waffles or cereal. You know, these products, these foods are made with wheat, and wheat is a plant product. There's lots of carbohydrates in these breakfast foods, sugars specifically, and sometimes in the form of starch. Starch found in flour is made up of glucose molecules linked together in one long chain. And your body has enzymes, once you eat that food, to start to break apart those glucose molecules, breaking the bonds that hold all of them together, releasing the glucose molecules so that they can be entered to your bloodstream, and your cells can start to oxidize it. So take a look, right? How does respiration start? It starts by you eating food. You would digest your food. You have enzymes that help to break down that food into glucose, and then it enters and dissolves into your bloodstream. Our body has a way of regulating the amount of glucose, sugars, within our blood. And that happens with a hormone called insulin. When glucose levels increase in our blood, our pancreas makes more insulin, which acts as a key for the glucose to enter into the cells. Now, the glucose, it's too big to just dissolve past the cellular membrane. It doesn't work like that. So the glucose has to enter our cells through glucose transport proteins. And they're closed normally. So in the presence of insulin, these protein channels will open up and glucose will enter into our cells, where the very first step of cell respiration occurs. That very first step, it happens in the cytoplasm of our cells, plant and animal cells. It happens with glycolysis. Glycolysis means to break down or split glucose. Glyco refers to sugar. Lysis means to split. So glycolysis is the splitting of glucose. 4 ATP are made, but it takes a small investment of energy, 2 ATP, to make the 4. So we say that the process nets 2. Notice that at the end of the process, so glucose is a 6-carbon molecule. Enzymes, a little bit of energy, will break apart glucose, cleaving it into 2 pyruvates. And so if you notice, we went from 6 carbons, 1 molecule with 6 carbons, to 2 molecules with 3 carbons each. We make a little bit of ATP, but we'll be able to break apart and strip the electrons off of those pyruvates throughout the process, utilizing that energy to make different forms of chemical energy called ATP. So this is where the process happens. Actually, the cytoplasm here is all of the areas outside of these circular membranes. So that's the location of the glycolysis process, right? But once we have made 2 pyruvates, additional enzymes will break it down into something called acetyl-CoA, which is a 2-carbon organic compound. That will enter into the mitochondria. And if you look here at these interesting mitochondria, there's an inner membrane and an outer membrane. You're seeing the boundary of the mitochondria, the outer membrane. But if you look really carefully inside, all of these structures are called cristae. That's actually the inner membrane itself. And all of those folds are actually increasing the surface area inside the mitochondria. Why is that important? Because what's about to happen next, electron transport chain and ATP synthase, can all be found within these inner membranes of the mitochondria. So if the mitochondria have additional folds in there, there's all that much more surfaces in order to undergo these processes. So let's do a quick little overview here. We've talked about step 1, glycolysis. Glucose is broken down into 2 pyruvic acids. These pyruvates are additionally modified by enzymes in the cytoplasm and transform them into 2 carbon molecules called acetyl-coenzyme A, or acetyl-CoA. Think about what happened. When we went from 3-carbon pyruvates to 2-carbon acetyl-CoAs, one of our carbons actually exits the system in the form of carbon dioxide. that we actively, as humans or animals, actively breathe out, right? Hey, this acetyl-CoA will enter into the mitochondria. During this process, additional enzymes will seek to rip apart that glucose, oxidize it, break it down, and strip those electrons off. So think about it. We've got 2 acetyl-CoAs. One turn of the Krebs cycle will reduce the carbon in these molecules down to 1. An additional turn of the Krebs cycle, and the carbons have now exited the system in the form of carbon dioxide. This is step 2 of the Krebs cycle, or is the Krebs cycle. We make a little bit of ATP here, and we also make some carbon dioxide as glucose is being oxidized, entirely broken down in this process. Now, what happens to its potential energy? The potential energy as we break apart glucose is being held by these high energy electron carriers, NADH and FADH2. In fact, a little bit of NADH is produced during glycolysis and during the modification of pyruvate to acetyl-CoA. These high energy electrons will move down into step 3, which is electron transport chain. Electron transport chain proteins in the inner membrane pass those electrons from one another, and as they do that, they pump H+ ions into the inner membrane space. That's the area here in the lighter orange of our mitochondria image. At the end of the electron transport chain, oxygen, if it's present, will accept those electrons at the end of the electron transport chain. When that happens, it reacts to form water. H2O is produced here during this process. In step 4, the H+ ions right up here in this lighter orange color of the mitochondria flow through that protein ATP synthase and produce ATP. And water, like I said, at the end of electron transport chain, that H2O is produced when electrons accept, let me say it again, when oxygen accepts the electrons at the end of the electron transport chain. So let's get some of the details here so that we can put this into our notes here for these 4 steps that we've outlined. Step 1, glycolysis. This is all being facilitated by enzymes. These structures that you see on your screen represent the enzymes involved with these different processes. So, and they're doing a lot of the work, right? A little bit of energy is used in order for enzymes to split glucose into 2 molecules of pyruvate. This happens in the cytoplasm of the cell, so we're not even in the mitochondria at this point. In order to be able to split from a 6-carbon sugar into 2 3-carbon pyruvates, a little bit of energy is invested. And it uses this process uses 2 ATP molecules to make 4 ATP molecules. So we say that it nets 2. We make 2. We increase the system by 2 ATP. So those pyruvates then will be modified into acetyl-CoA. So we're going to strip a carbon off of those pyruvates and transform them into acetyl-CoA. And when we do that, we produce a little bit of carbon dioxide, sure. So pyruvate is modified into acetyl-CoA, which is oxidized, further broken down. Its high energy electrons are held by NADH and FADH2, these high energy electron carriers. In a moment, they'll shuttle the high energy electrons to the electron transport chain, but we'll get to that. This process is happening inside the mitochondria. So as we continue to oxidize glucose, it's happening in the matrix of the mitochondria, on the inside. This process makes a little bit of ATP. Like glycolysis, we make 2 ATP, but we're not done yet, right? We're making a lot of carbon dioxide as well. But those high energy electrons are going to help to make the bulk of the ATP, which is about to happen here in steps 3 and 4. So step 3 in our diagram is the electron transport chain. So those high energy electrons that were transferred to NADH and FADH2 during the Krebs cycle are going to be passed along these protein chains here, the embedded proteins within the inner membrane of the mitochondria. The electrons will pass down from one of these proteins to the other, and as that happens, these proteins are going to actively pump H+ ions into the inner membrane space, which is in between the 2 orange cellular membranes. We're increasing the concentration of the H+ ions in this section or this region of the mitochondria. Hey, something that's interesting that happens here is that one of these proteins at the very end of the electron transport chain will take oxygen gas that you're actively breathing in right now. Think about what's going on. Oxygen gas, and oxygen is going to accept the electrons at the end of the electron transport chain, combine with some H plus ions in the area and form H2O, water. So you guys are actively producing water during this process. And if you don't believe me, go into your bathroom in the morning when it's nice and cold and blow on the mirror and you'll see condensation. That's the H2O, that's the water that you're producing from this process. Now listen, if you don't have oxygen gas, you don't have a molecule that will accept the electrons at the end of the electron transport chain. So if you don't have the electrons moving out of the system, everything, because oxygen isn't present, this process will grind to a halt. The electrons stop moving, we stop pumping H plus ions into the inner membrane space, and we'll stop ATP production. So I'm going to encourage you guys to remember to keep breathing. Keep breathing, because that oxygen is very important. That oxygen is the final electron acceptor in an electron transport chain. And without it, if we weren't making water, this process, the passing of the electrons down the electron transport chain, stops. And that becomes an issue. You'll very likely die if you don't start breathing again. So just remember, breathe the oxygen in, use it, allow the oxygen to accept the electrons at the end of the electron transport chain, and then breathe out the carbon dioxide being produced during the Krebs cycle. All right, and then step 4, that brings us to oxidative phosphorylation, which is a fancy way of saying the production of ATP. We're phosphorylating ADP, adding a phosphate to it to make it ATP. It has a little bit more potential energy. That third phosphate helps us do some work. And so what happens then, if we've increased the amount of H plus ions in this inner membrane space, that H plus isn't able to pass from an area of high concentration to an area of low concentration. It just can't pass through this region. This cellular membrane does not like positive ions. It doesn't like charged particles. So, but there is one location for that H plus ion to move through, and that's through this protein here embedded in the inner membrane space, ATP synthase. What this ATP synthase is able to do is harness the movement, the kinetic energy of the H plus ions as it moves through, and attach a phosphate to make ATP. And this process, guys, will make 28 to 32 ATP. So if you count up the number of ATP in glycolysis, 2, the number of ATP produced by the Krebs cycle, 2, and in the process here, oxidative phosphorylation, 28 to 32, we're looking at 34 to 36 ATP molecules. Some textbooks might go all the way up to 38, but we're not always that efficient at the total oxidation of glucose. There's genes involved. There's different reasons why we might have a slower metabolism. So we're not always making all of that ATP, in a range of 32 to 36 or so. All right, so to recap, we've got glucose. Enzymes break glucose, split glucose into 2 pyruvates. These pyruvates are additionally modified into acetyl-CoA. And when that happens, we release carbon dioxide out of the system. The acetyl-CoA in step 2 will go through the Krebs cycle and release more of that carbon dioxide out of our system. The electrons that were initially found on acetyl-CoA are now going to be carried by NADH and FADH2, these high energy electron carriers. In step 3, electrons will move down the electron transport chain, pumping H plus ions into the inner membrane space. Those H plus ions will move through ATP synthase, oxidative phosphorylation will occur, and we make a lot of ATP. As long as oxygen's present, right? Hey, we also make water, because when those electrons react with the oxygen at the end of the electron transport chain, it produces water. If O2 wasn't present, everything stops. You don't make ATP and death is imminent. So I have this, what I'm going to do next session with you guys is go through and let's do the respiration dance and have you guys engage with this material a little bit. So I've got these lyrics and some dance moves that I'd like to teach you. And if you watch the video at crjust.us/RD for respiration dance, you'll see the video tutorial that I created for that. In fact, this link to the respiration dance and lyrics and the dance moves can be found on our Canvas class. I hope this was helpful. Let me know if you have any questions.